Hello, my name is Matt Guart, and I am presenting the synthesis of isoxazoline and isoxazole heterocycles as potential inhibitors of lysyl oxidase. So the enzyme lysyl oxidase, or LOX, is one of many proteins in a family of copper-dependent amine oxidases best characterized by their function to cross-link elastin and collagen proteins. A key detail is that the enzyme's regeneration, or catalysis, is dependent on a lysyl tyrosylquinone, or LTQ cofactor, which participates in the conversion of lysine amine side chains into aldehydes and ultimately results in the cross-links between collagen and elastin, as shown in the mechanism to the right. Since oxygen is required for this process to occur, LOX is noticeably upregulated while in an oxygen-poor or hypoxic environment. It is known that the overexpression of LOX is prevalent in hypoxic tumors and promotes the metastasis of many cancers, but most often this occurs in breast and colon cancers. Inhibitors such as beta-aminopropionitrile, or BAPN, seek to react with the LTQ cofactor at the active site of the enzyme, resulting in irreversible binding and enzyme deactivation. While direct implementation of BAPN is logical, its toxicity would effectively carpet bomb the entire organism and likely yield more physical damage than cancer retreat. Thus, prodrugs that release BAPN directly to cancerous tumors are highly sought after. Both the Solano and Lopez labs at CSUB have worked in conjunction to synthesize and assay targeted LOX inhibitors, but despite successfully synthesizing them, they have suffered from limited water solubility and therefore had limited drug bioavailability. Thus, this prompted additional research to produce inhibitors with increased hydrogen bonding and therefore water solubility by including heterocycles within their structures. Heterocycles are ubiquitous in many realms of industry, and because of their prevalence in biological molecules, these ring structures are highly sought after, and as a result, over 80% of best-selling small molecule drugs contain at least one heterocycle within their structures in the US 2010 market. The nitrogen and oxygen heteroatoms in rings like isoxazoles and isoxazolines provide excellent binding sites to protein targets with broad ability while also adding hydrogen bonding donors and/or receptors that increase aqueous solubility as mentioned previously. Previous heterocycle research has primarily focused on the development and optimization of a one-pot greener method of synthesizing isoxazoline derivatives using recyclable surfactant. The surfactant TPGS750M, or TPGS, was specifically chosen for its promise of recyclability and formation of optimally sized micelles that serve as an aqueous reaction microenvironment. In doing so, this synthetic route has allowed successful production of isoxazoline and isoxazole products to be produced simply and earth-consciously with room for modification, such as in the case of our research proposal. The full development and optimization of this scheme offers an obvious partnership with inhibitor synthesis by using these novel heterocycles in aim to increase aqueous solubility, potency, and ability of future inhibitors. Because of its potency in previous inhibitors, 4-bromobenzaldehyde was chosen as the base for the flagship isoxazoline inhibitor and later extended to the isoxazole variant as shown below. It is important to note that as in most chemical syntheses, each molecule in the scheme that is noted with a percent yield represents a full synthesization, purification using column chromatography, and characterization by nuclear magnetic resonance, or NMR. To begin the synthesis, 4-bromobenzaldehyde was condensed with hydroxylamine hydrochloride to afford the oxime. This oxime then underwent 1,4-dipolar cycloaddition with methyl acrylate and TPGS solution to yield the isoxazoline. After column chromatography, a yield of 53.2% was obtained and the molecule was fully characterized using NMR. In order to arrive at the final inhibitor molecule seen prior to this, a final amination with the BAPN must occur, thus the ester product was required to be reduced to a more reactive aldehyde product. Direct partial reduction of the ester to the aldehyde was ideal in order to conserve on redox economy and overall efficiency. Thus, diol, or diisobutyl aluminum hydride, was employed as per the literature, but consistently over-reduced the ester to the alcohol. To work around this, complete reduction to the alcohol and partial oxidation to the aldehyde was proposed in order to continue the synthesis as shown. Reduction using sodium borohydride was successfully used to reduce the ester and yield a 92.4% conversion. Although this was successful, oxidation was not so straightforward. Manganese dioxide was first thought of since it offered a facile and green partial oxidation, but as it was later discovered, a nearby allylic or double bond was needed for the alcohol's OH group to be oxidized. As a last resort, the Swern oxidation was attempted per the literature as shown, but failed miserably due to the alcohol's insolubility in the dichloromethane reaction solvent. While not ideal, the target molecule was entirely shifted away from the isoxazoline to the isoxazole due to the hope that the manganese dioxide would be able to oxidize its allylic alcohol, unlike the isoxazoline's absent one. The entire reaction methodology up to this point was extended to the isoxazole variant as shown. The starting molecule was made in exactly the same manner as the isoxazoline, except for the replacement of methyl acrylate for methyl propiolate. Reduction of the ester to the alcohol and subsequent purification gave 64% yield. After manganese dioxide was successfully confirmed to oxidize the alcohol, and inspired by what some research groups had reported, a one-pot partial oxidation with manganese dioxide and condensation with an amine was extended to our alcoholic substrate to yield the imine, which was then reduced to the amine product shown. After purification, a yield of 43.4% was obtained, and the product was fully characterized by NMR, as you can see to the right. Black, orange, blue, and green circled peaks are color-coded and represent the hydrogen atoms present in the benzene ring, isoxazole, the BAPene linkage point, and the chain's entirety, respectively, according to the molecule shown here, proving that the target molecule was successfully synthesized. In total, the combined yields represent a 27.8% yield for the final isoxazole inhibitor. This value, along with the NMR data, provides both quantitative and qualitative observations, which will be considered when attempting to scale up the reactions for the bioassays Dr. Lopez's group will establish. Although the isoxazoline inhibitor was unable to be produced, it is expected that the manipulation of the Swern reaction protocol will allow for it to be made soon, along with a future library of isoxazoline and isoxazole inhibitors. I would like to recognize Drs. Daniel Solano and Carlo Lopez for their unwavering patience, guidance, and mentorship throughout this and future research I conduct at CSUB. I would also like to thank Dr. Zelezny and her associates for providing this exceptional undergraduate research experience. Thank you for your time.